Food preservation. When food rots, it becomes a fertile growing medium for microorganisms. The microorganisms break down the nutrients in the food, destroy its flavor, and release toxic byproducts that make people sick. Preserving food prevents the growth of microorganisms on it before it is consumed. It is preserved by refrigeration, heating, drying, salting, and then fermentation, all different methods. Biochemical reactions, like all chemical reactions, require activation energy to proceed. Molecules must collide in order to react, and enzymes need to be able to change shape in order to work. As the temperature increases, the molecules collide more, and enzymes become more effective, increasing the rate of reaction for microbes. The microbes reproduce faster and grow on the food. Refrigeration lowers the temperature, so these microbial enzymes can no longer catalyze biochemical reactions as there are fewer collisions, and the result is the growth of microbes on food is inhibited. It's slowed down. The graph shows that the growth rates of different strains of E. coli, a very dangerous bacteria, increase as temperature increases. So our temperatures down here, 5 Celsius, 15 degrees Celsius, all the way up to 45 degrees Celsius. And this is the growth rate. So at very low temperatures, it has a very small growth rate. The growth rate of the microbes, of the E. coli, increases right until you get to about, 43, maybe 43 degrees Celsius. And then, when it's heated even more, then it starts dropping off. And we're going to talk about that shortly, which is why we heat our food. But back to refrigeration. Food stored at 2 to 4 degrees Celsius in the refrigerator will grow mold, fungus, and bacteria very slowly. Below 0 degrees Celsius, biochemical reactions cannot proceed because molecules cannot move, so microorganisms cannot grow. Food will last a long time in a freezer. The amount of time that a food remains edible at room temperature, refrigerated, or frozen varies depending on the type of food. Heating. At high temperatures, proteins unfold and denature. Now let's just revisit that graph earlier, the E. coli. We saw that the rate of growth keeps increasing with increasing temperature, but at a certain point, it starts falling off again, just as when we refrigerated food. So, for heating, when this happens to microbial enzymes, they can no longer catalyze biochemical reactions. That's why food is cooked at high enough temperatures and long enough to kill microbes. That's over on this part of the chart. Milk and cream are heat pasteurized, heating them to temperatures that kill microbes and then packaging it using the sterile technique. Pasteurization greatly increases the shelf life of dairy products. Here's a little mnemonic. When caring for food, keep hot foods hot and keep cold foods cold. Foods that have been heated but then left at room temperature for hours will begin to grow microbes. Hot food should be consumed within a couple hours of being heated and kept hot or then refrigerated. Foods that have been refrigerated need to be kept at cold temperatures. If left at room temperature long enough, food will spoil. Now let's move on to drying. Cellular compartments are filled with water and ions, and almost all biochemical reactions take place in water. The water inside cells helps them maintain their shapes against outside pressure. Shape is important at the molecular level for biochemical reactions. Water is a participant in the reactions that are required for life. Very key statement right here. Such as photosynthesis and the breakdown of larger molecules for cell use. Its polar properties are a key factor in the creation of cell membranes. Drying food removes the water required for microorganisms' metabolism. They cannot grow or survive in the absence of water. Therefore, drying foods preserves the food without removing the calories and other nutrients. Here's how fish can be preserved. They're drying in the sun and the water is being removed. It's evaporating away from them. Salting, which is actually a form of drying. First, osmosis is the movement of water across a semi-permeable membrane from a lower concentration of solute to areas of higher concentration of solute, and in this case salt will be the solute. Covering food with salt results in a higher concentration of salt outside the cells than inside the cells. The cells are full of water but little salt. Therefore, the water will move out of the cells where there is less solute, or salt, to the added salt, which is more solute, outside. The cells dry out. As water is removed, biochemical reactions stop in the microorganisms as discussed in the previous slides, and the food is preserved. And here's another way to dry out fish. They're covered with salt, and that will help prevent them from spoiling. Brining is another type of food preservation using salt. Brining involves soaking foods in a solution with a high salt concentration and an acid such as vinegar. Brining vegetables is also called pickling. Pickling can make foods easier to digest and add flavor in addition to preserving them. Fermentation is the alteration of food by the controlled use of bacteria or yeast to enhance its shelf life, flavor, and/or nutrition. Fermentation is anaerobic cellular respiration. Microbes can carry out fermentation using food as a source of glucose or other carbohydrates. We have two types. One is alcoholic, where glucose is converted to ethanol and carbon dioxide. Then we have lactic acid fermentation. This time, the glucose is being converted to lactate. In this dough, yeast, which is single-celled fungi, is converting carbohydrates to carbon dioxide and ethanol. The carbon dioxide causes the dough to rise and bubble. The ethanol evaporates when the dough is baked. Yeast can be made by mixing water and flour, such as sourdough bread, or just using baker's yeast, which is this word over here. Alcoholic fermentation of grains or fruit carried out by yeast is used to produce beer and wine. The production of ethanol in beer and wine greatly increases the shelf life of those beverages because it inhibits the growth of microbes. Some alcoholic beverages are fermented by naturally occurring microbes, while others are fermented by selected yeast cultures added to the food source. This person is making thabwa, or sweet beer. It is made with fermented maize and millet or sorghum flour. After boiling and cooling, natural lactic acid producing bacteria work on the porridge for 2 days in clay pots before the drink is served. Longer fermentation times allow yeast to grow, which turns the beverage to alcoholic beer or moa. Lactic acid fermentation of milk and cream, carried out by bacteria such as lactobacillus, produces yogurt, kefir, or chambiko, or other sour dairy products. Natural environmental strains of bacteria carry out the fermentation, and in other cases, specific lactobacillus cultures are added. The food source for the lactobacillus is the lactose sugar present in milk. The bacteria convert lactose to glucose and then to lactic acid. The non-alcoholic fermentation of vegetables and fruits involves soaking them in a salt concentration that allows only the growth of certain salt-tolerant bacteria. The salt concentration in fermentation brine selects which microbes grow and inhibits unwanted bacteria, mold, and fungus. Fermentation prevents food spoilage and enhances flavor. All fruits and vegetables have microbes that can help carry out fermentation. The type of bacteria that grow in the presence of salt are lactic acid producers. The fermenting bacteria enhance the food in several ways. Lactic acid adds a tart flavor, and other byproducts of bacteria add complexity to the flavor. The bacteria make more nutrients available for digestion as well by breaking down some of the cellulose, and that's the complex carbohydrate that makes up cell walls and that humans cannot digest. So we have cellulose, which is undigestible, turns into glucose, which is digestible, and also lactate.